Okay, so I was going over this and then my voice went robotic. So I'm recording again. But I was just mentioning that I went through the first 5 points. And then I got to Vmax and Km. We mentioned them briefly last time. I'll talk more about them now when we start talking about kinetics. And we talked about the importance of enzyme kinetics, understanding the parameters so that we can design our either enzymatic reaction in the lab or enzymatic processing. So we really need to know the parameters to set up our enzymatic assay or process. So yeah, I mean, I was talking about the induced fit query and how the environment impacts the active site, and then how the active site changes to allow the substrate to bind. So it's not a rigid lock and key. And also here is showing the Vmax and Km. So the Km here is for 2 substrates and 1 enzyme. And the 1 enzyme has higher affinity to the red dotted line substrate because it has lower Km. That means you reach Vmax over 2 at lower concentration. So this means you have higher activity or affinity of the enzyme to the substrate. And so Km is the concentration of the substrate at Vmax, Vmax, or half Vmax. All right, Radhika, do you have any questions? Okay, moving on. So we start off talking about the time course of an enzymatic reaction. So when we're setting up our enzyme kinetics assay to determine Km and Vmax, we start off by looking at plotting the product concentration against the time of the reaction. So what happens at the beginning of the reaction, few seconds it takes for the substrate to bind to the enzyme, and this is the pre-steady state over here. But then what we care for is the steady state here, which is almost linear. So with more time, you have linear increase in product. And at this steady state, we should remember that rate of disappearance of enzyme substrate equal rates of appearance of the enzyme substrate complex. So if you look here at this reaction, so you have 3 rate constants that you see. So you have the first rate constant, which the enzyme and the substrate forming the ES, and then the second rate constant where the rate of disappearance, so the ES convert the substrate to product and releases the enzyme. And then you also have a backward reaction. So you have a rate constant that actually the ES dissociates into E plus S as well. So this is the kind of the essence and the basis of what we're going to talk about next in terms of deriving the Michaelis-Menten reaction that or equation that we use to determine Km and Vmax. So at the steady state is when we really want to monitor our reaction, and the slope of that steady state here, or the linear portion of the curve, is the initial velocity of the reaction. So as you can see here, as time goes by, the linear reaction kind of starts to plateau and just stop increasing. Why is that? Because you have basically you start depleting the substrate, and when the substrate starts depleting, then you lose the linearity of your reaction. So monitoring reaction parameters has to be during the steady state. So during the time period where you can actually get a linear line and where you can get the slope, which is the V0 or initial velocity of the reaction. So again, we really need, the purpose of all of this is to get the kinetic parameters. So in order to get the kinetic parameters, usually you set your reaction at different substrate concentration. So you repeat the enzymatic reaction, you keep the enzyme constant, the enzyme concentration constant, and you repeat the reaction at different substrate concentration. And at each substrate concentration, you get the initial velocity. And then let's say you did 5, 6, 7 concentrations, you plot the velocity, initial velocity at each concentration over the substrate concentration. And you obtain this graph, which basically also it starts linear. And at the beginning here, when it's linear, it's first order with respect to the substrate. So the velocity is dependent on substrate concentration. And then as we go up, or as we increase the substrate concentration, we reach Vmax and we don't have any more increase in velocity because the enzyme is saturated with the substrate. So no matter how much you increase the substrate, there's not going to be an impact on the rate of the reaction. You just maximize the rate of the reaction at that particular enzyme concentration. So you have one constant enzyme concentration, but when we increase the substrate and the enzyme is saturated, that's it. We reach Vmax, no more. So Km again is here the concentration of the substrate at which you have maximum velocity. So that is what I just explained about reaching Vmax. And then the Km is definition, the actual definition is the concentration of the substrate at half Vmax. But what does it mean really? It tells us the affinity of the enzyme to the substrate. So the lower the Km, the higher is the affinity. That means at lower substrate concentration, you reach Vmax over 2. Okay, so that means the enzyme have high affinity to the substrate, it binds, it reacts quickly at lower concentration. Okay, so what are these two important parameters? What are they impacted by? So if we look at Vmax, it's impacted by the nature of the substrate as well as the concentration of the substrate. The enzyme concentration, the higher the enzyme concentration, the higher the rate of the reaction. Of course, environmental conditions, the pH, the temperature, because that impacts enzyme activity. And if you have inhibitors or you don't have, and the type of inhibitors, which I will explain here later on. So Km is affected also by the nature of the enzyme. Obviously, different enzymes have different affinity to specific substrates. And if we're talking about one enzyme, different substrates. So the enzyme has different affinity to different substrate, and hence this figure here that I showed earlier with two substrates, one enzyme, and the same enzyme has two different Kms to the two different substrates. And of course, changing the pH or the environment conditions such as temperature, even ionic strength, will impact the affinity of the substrate, the enzyme to the substrate, because of its impact on the active site. And definitely types of inhibitors. We will see that some types of inhibitor impact directly Km, other types of inhibitors impact Vmax, and we'll talk about that. So if we want to put it in context, what does it mean to have a good consequence of low Km? When is it good to have low Km? Low, that means high affinity of an enzyme to substrate. When we're using the enzyme for production. So for example, we're producing corn syrup. So we're using an enzyme, amylases, to produce corn syrup. So we really want to have low Km. This means we have a high production at lower concentration of enzyme, of substrate, basically. So we produce more in shorter period of time. Yeah, basically that's what it is. So if we want for two enzymes here that react on the same substrate, to produce the same desirable product, we choose the enzyme with the smaller Km, okay? Because this is going to give me better yield unless the substrate concentration are not limiting and really high. So it is, so yeah, my daughter is distracting me. Sorry, she's texting. Anyway, so yeah, so if the concentration of the substrate is high, it really doesn't matter. But when the substrate concentration is not that high, then we choose the Km or the smaller Km. So when is the consequence bad of low Km? So when we don't want to have a low Km is basically when we have an undesirable enzyme reaction going on, like polyphenol oxidase and browning. So we really don't want to have a low Km of the enzyme polyphenol oxidase to the substrate that we have. So because of at low substrate concentration, we're going to get high rates, and then this means, like in polyphenol oxidase case, high browning. Okay, so a couple of things that we need to remember, and I usually ask about, is when your substrate concentration in a reaction is much lower than the Km. So in this region of the graph, here we have first order with respect to the substrate. That means when we change the substrate concentration, the rate of the reaction is impacted. Okay, so in this case, we set up our experiment in this case when we are determining constituents concentration, like glucose concentration. So I want to make sure that my glucose concentration in the enzyme assay that I'm following is much less than the Km. So I really need to know the Km of the enzyme towards this particular substrate so that I set up my assay in a way that I can detect change in concentration. And that's what I want, because I want to measure concentration in unknown samples. Okay. However, on the other end, if I care about measuring enzyme activity, I want the assay or the rate of the reaction to be dependent on the enzyme concentration or enzyme activity. I don't want it to be dependent on the substrate. So I want to set up my reaction where I have a lot of substrate. So I saturate the enzyme, so it's zero order with respect to substrate and first order with respect to enzyme. That means if there is a change in enzyme concentration, I can pick it up based on the rate of the reaction. So when I'm measuring enzyme activity, I need to work with substrate concentrations much greater than Km so that they are not impacting the rate of the reaction, but the enzyme concentration is. Okay, so the Michaelis-Menten equation is very important because it is the equation that leads me to calculate Km and Vmax. So I'm going to walk you through derivation of this equation. So what parameters are needed to determine these parameters? So the very first thing that we start with is what's the initial velocity in terms of an equation. So if I look at the velocity, really, it's the velocity is the rate of the production of product over time. So this is your slope. So the V0 is the slope here. And that is dependent on the concentration of ES, which is the complex of enzyme and substrate, and the rate constant. So initial velocity equal the rate constant that gets us to products multiplied by the concentration of ES, because this is directly related to the production of product. Okay, so we have that. And the other thing that we know also at this steady state, we have something called the rate of appearance of ES. So the rate of appearance of disappearance of ES. So we talked about the ES converting to product. and release the enzyme. And we talked about the ES also going backwards, dissociating into E plus S. So I have two rate constants. And so the disappearance of ES is a combination of these two rate constants. So it is the rate constant of going backwards multiplied by the concentration and the rate constant going forward multiplied by the ES concentration. So that's rate disappearance. The rate appearance of ES is this just one, its appearance going that way. So it's this rate constant here multiplied by the concentration of the enzyme and the concentration of the substrate. So during the steady state, rate of disappearance equal rate of appearance. So this basically here is equal that. So I want to reduce the number of different factors I have. So what do I know? I know that the total concentration of enzyme equal the enzyme, the free enzyme basically, and the ES complex. So the concentration of the total enzyme equal the concentration of the free enzyme plus the concentration of the enzyme substrate constant, the substrate complex, sorry. So if I want to replace E in this equation and have ES there, that's what it would be. So I would replace E. It would be total enzyme concentration minus total concentration of the enzyme substrate complex. All right, so now I'm going to solve for ES. So making some rearrangements here in the equation and solve for ES concentration. Again, I am trying to get to this V naught equal K2 into ES. So I try to see what the ES concentration is by just reassembly of the equation and solving for ES. I get this equation, which is on the next page, the same equation. So these constants, reaction constant, this K minus 1 plus K over plus 2 over K1, this is Km. So this is the Michaelis-Menten constant. So now my ES equal the concentration of total enzyme, concentration of the substrate over the concentration of substrate plus Km. So what does this, how does this relate to here? So basically, if I want to replace ES in this equation, I get the initial velocity here, the Km is here, and I have this K2 into the total concentration of the enzyme. All right, so I'm putting that on the next page. It's the same equation, but what is Vmax? Vmax is reached when the enzyme is saturated. That means all of the enzyme is in the ES form. So when all of the enzyme is saturated, we get Vmax. So Vmax, so basically you have K2 into ES equals K2 into total enzyme. So all of the enzyme is in the form of ES. Therefore, Vmax now equal K2 into the concentration of all of the enzyme. Because in this case, we reached maximum velocity, all the enzyme is saturated. So all the enzyme is in the form of ES. All right, so now this here function, K2 into E naught, or total enzyme, represent Vmax. So now I have an equation that has my initial velocity that I can calculate from the slope of the reaction at one concentration. Then I have the concentration of the substrate, which I know. And then I have the Vmax and Km that I want to calculate. So this equation here combines all the factors that I either can calculate or is known, and the two factors that I need to get from relationship from either a linear relationship or a nonlinear relationship. This is the Michaelis-Menten equation, but this here, it relates to this whole graph, which is nonlinear. And usually we like to work with linear so that it's easier. We have a slope, we have an intercept, so we can get parameters that we're looking for. So this equation here that has all of the parameters I care about, if it's inverted, it becomes a linear relationship, which is known as the Lineweaver-Burk plot. So basically, this equation right here is inverted. So instead of V0, it's 1 over V0, the inverse. And instead of the concentration of the substrate, it's 1 over the concentration of the substrate. So we end up with equation of the linear line, which is y in this case, equals the AX, X is 1 over the substrate, plus B, which is the intercept. But here's what we have. 1 over Vmax is the intercept. So I can calculate from the equation of the line, I'll get the intercept, I can get Vmax. Great, I got my first parameter. So, and then the slope is Km over Vmax. Then if I have Vmax from the intercept and I have the slope of the line from the equation of the line, I can calculate Km. So now I got my parameters that I'm looking for to understand the relationship between this enzyme and this substrate. So again, how do I get the V0? Is I run the equation, or I run, not the equation, sorry, the assay at different substrate concentrations. So I run my assay multiple times and I get the slope here. So I have the product produced over time. I get the slope of the reaction, that's V0. I do that 5 or 6 times at different concentrations, 5 or 6 concentrations. Then I plot the inverse of the initial velocity at each concentration and the inverse of each concentration. When I plot it, I got the line, I get the equation of the line, and from the equation of the line, I can get Vmax as an intercept, and then from the slope, and I have the Vmax, I can get K. Okay. So I have my parameters. All right, this is an example of applying the Lineweaver-Burk plot. This is a project I worked on as a postdoc, and in that project, I was looking at the bioaccessibility of 3 different isoflavones, phytochemicals in soy. So all of these 3 isoflavones has a sugar moiety attached to them. In order to be bioaccessible or absorbed into our intestine and be able to utilize them for the benefit, the sugar moiety has to be removed. So there's an enzyme in our gut called glucosidase enzyme that removes that glucose sugar and allows that molecule to be bioaccessible. So we were looking at which of these 3 are more biologically available, bioaccessible, based on how well the enzyme hydrolyzed the glucose off of them. So we designed an experiment, we ran it at different concentrations, and we determined the Km and Vmax for each of these substrates. So the enzyme towards each of these substrates. And we found out this triangle here represents the glycetins. It has the highest slope. So that means it has the higher Km. And the other 2 are very similar. They had very similar Vmax, the intercept. They have very similar intercept and a much smaller Km. So basically, we found that the enzyme towards this particular compound, glycetin, had low Vmax and low Kcat over Km, which is catalytic efficiency, basically. It's another term we calculate. And Kcat is Vmax over total concentration of the enzyme. So the lower the Vmax, the lower the Kcat, the lower the catalytic efficiency, and the higher the Km, also the lower the catalytic efficiency of that enzyme towards the substrate. So we determined that these two phytochemicals are more bioaccessible than this one. Okay? So that is one application. We'll give you more examples. Okay, so factors now that affect the enzyme reaction at high concentration of substrate. Okay, so when the substrate is saturating, the enzyme system is high in substrate, then as you increase the enzyme concentration, the velocity increases. So that becomes first order with respect to the enzyme. So the higher the enzyme concentration, the higher is the rate of the reaction. So, however, we need to determine when we're setting our assay, we need to determine the concentration of the enzyme that we want to use and the time that we want to use to monitor our reaction or to end our reaction. So these two factors are important. So notice here what happens. So this is enzyme concentration 1, enzyme concentration 2, 3, and 4. So with each one, we see a higher initial velocity because this is the slope, right? So you have a higher slope with the higher concentration. However, look at the prediction line versus the actual. What's happening is you're depleting the substrate fast, so you're getting plateau, and then you are only linear in this range where probably time is like 1 minute, 30 seconds. It's not enough time to control the reaction and be able to stop it and do the analysis. So you don't want to work with a reaction where it's so fast that you cannot control it. So you want to select a time that, let's say, 10 minutes, 15 minutes is reasonable time. And you want to select an enzyme concentration where at this time point, your prediction line is linear and equal to the actual, okay, adjacent to the actual. So you don't want to deviate from the actual. You don't want to deplete your substrate quickly so that you lose linearity. You want to be in this linear portion. So potentially, instead of enzyme 1, we can go with concentration 2 at a reasonable amount of time and good, accurate prediction of the velocity or initial velocity. So this is really important. And this is, again, it shows you here that you want to be in linear portion. You want to be in the steady state. You do not want to deplete. You don't want to rate a high rate of reaction that depletes the substrate and plateaus like this here, where your prediction is not linear. You're not in the steady state. Okay, so you have to be in the steady state, and that is important to determine time and enzyme concentration. So other than enzyme concentration, substrate concentration, and we talked already about saturating the enzyme where you get maximum velocity. So we talked about first or zero order. But there's also, if the substrate concentration is really high, sometimes it causes inhibition. So how does that work? So if you have a lot of substrate and not a lot of enzyme, so what happens, sometimes the substrate bind at a different location on the enzyme, changes the active site, so changing the configuration of the enzyme. So the active site changes and no longer is efficient in binding the substrate at the active site. So we call that enzyme inhibition. Sometimes you need substrate activation, where actually the enzyme binds at a different location and enhances the active site in a way, changing its structure in a way that it has now better affinity to the substrate. So these are the 3 main effects. Environmental factors, temperature effects. So here, we all know that with increase in temperature, we have higher thermodynamics. So we get higher velocity. You have more movements, you have more interactions. And you get maximum velocity at a specific temperature that is optimal for each enzyme. So this temperature is not always 37 degrees C. It's not always 60 degrees C. It depends on the enzyme. So it depends on the enzyme. You might get increase until you reach the optimum. It might plateau, decrease a little, but as soon as you hit denaturation temperature of the enzyme, because the enzyme is a protein, it will denature if you keep increasing the temperature. The enzyme will denature and the activity will decrease, basically. We start high, and then once the protein is denatured, the enzyme basically loses its activity. So there is, and this happens at different temperatures for different enzymes. In terms of pH effect, we also have an optimum pH. Why do we have an optimum pH? Because basically you need the right charge for the active site to bind to the substrate. So you need to have the right ionic state, basically, of your active site to bind. So, and this is here example is pH 6, but it's not always pH 6 to all of the enzymes. Some enzymes have, like pepsin, has a pH of 1.3 to 2, where it's optimal. And another enzymes like alkylase, for example, so microbial enzyme, it has alkaline pH, which is 8, pH 7.5 to 8. So every enzyme has specific ionization state where it is most optimal. Okay, so when you go away from that optimum pH, the activity decreases. Now, there is a difference, though, between optimum activity and denaturation of the enzyme. So when you increase the pH a lot or decrease it a lot, the enzyme, like any protein, might be denatured and irreversibly denatured, so it won't act anymore. But then we talk about pH stability versus pH optimal. So for example, here you have trypsin. Optimum pH is 7 to 8, but it is still stable at pH 3. So you can lower its activity, but then if you change the pH back to 7 or 8, then you can get it back to be active again. The way to test enzyme inhibition or inactivation by pH is to bring the pH down to different pHs or up to different pHs and hold the enzyme at that pH and then bring it back to optimum to see if the enzyme is still active. If the enzyme lost activity, that means you kind of denatured it at that particular pH. Other environmental factors are activators. We have two types of activators. We have those that bind very strongly. They call it prosthetic groups. So once they're bound, they are not dissociated. You have a very low concentration or dissociation factor. So the other one, here's an example of like vitamin B6 is a prosthetic group to an aminotransferase enzyme. But most activators that we add to our system to enhance the activity of the enzymes are called coenzymes, and they dissociate, dissociate. So they're not, they don't permanently bind to the enzyme. And this is a common example of a dissociable coenzyme, which is NAD+. And this is used in the assay that we use to determine ethanol in a sample. So basically, the way we determine ethanol is we make sure that we have a coenzyme in the reaction that will activate alcohol dehydrogenase. So what happens here is you end up having the ethanol dehydrogenated to produce acetaldehyde, and you have NADH produced in the process, which absorb UV light. So this is how we determine ethanol. So NAD plus in this case is a coenzyme. Sometimes coenzymes are ions, like chloride ions, for example. So in some enzyme require a certain concentration of chloride to act. Inhibitors are a whole set of factors. We have two types, two major categories of inhibitors. I don't know what it did, it dropped something. So one is irreversible inhibitor category, and this means that it binds and it doesn't leave. Dissociation factor or constant is very small. So the decrease in enzyme activity in this case is directly related to the concentration of the inhibitor. Sometimes some inhibitors that are irreversible, it takes a long time for them to bind. So it's a slow reaction, but once they bind, they are irreversible. So for example, alpha amylase inhibitor, which is present in some beans and seeds, it is an irreversible inhibitor. So when we want to determine residual activity of amylase, let's say in the presence of an inhibitor, we want to make sure that we give them enough time to bind before we determine the activity, the residual activity of amylase. So you just have to incubate them for a longer time to get them to interact. So reversible inhibitors, the second category of inhibitors, we have three different types of reversible inhibitors. So we have a competitive inhibitor. So in this case here, let's say there's this enzyme. The substrate is succinate. It reacts with the enzyme, give you fumarate and the enzyme again. So this is the product. This is the substrate. However, another compound that is similar in molecular structure to succinate can inhibit this reaction. So look at malonate here. The only difference is a methyl group here. It has less methyl group. Somehow, with this very similar structure, it can bind to the enzyme competitively. So it competes for the binding site. And when it binds, no product is formed. This kind of competitive inhibition causes an increase in Km because it's competing for the same active site, then the enzyme affinity to the substrate is reduced. Just a second here. Sorry, I have. Okay, so let me show you the changes in Km. So in this example, you have your initial slope of the reaction without an inhibitor. And then once the competitive inhibitor was put in the system, the slope of that line increased, and the velocity did not change, but the Km changed. So the Km over Vmax became higher, an indication of increase in Km. So, but to overcome a competitive inhibition, you can increase the substrate concentration. At high saturated substrate concentration, then there will be no effect on the rate of the reaction in the presence of that inhibitor. Okay, so sometimes we want an inhibitor to be there. So a good competitor, your Km to the inhibitor is lower than the Km to your substrate. So that means the enzyme has more affinity to the inhibitor that you add. But once you have more substrate in the system, it will overcome that inhibitor basically. But on the flip side, sometimes you do not want to inhibit a certain reaction. So you really want to produce the corn syrup. You don't want any alpha amylase inhibitor in the system. So basically, you want to try to not have this undesirable competitor with lower Km than your substrate. In this case, is not desirable because it's going to slow down your reaction. Then you end up with this products convert or produced per hour. Therefore, you are forced to do something to remove the competitor by some means, or you want to increase the substrate concentration, basically.
Pam, I have a question
Yeah.
from the previous slide. Is there an example where we like to have a good competitor, industrial example or a research example?
So basically, yes. So for example, you have polyphenol oxidase. You want to put another substrate that would react. I give you an example later. That would react instead of the phenols that are in your whatever apple or any other produce. So you put an inhibitor to to stop that from reacting with the substrate that gives you browning. So you want a good competitor in this case. But if you are producing, like I said, a product and industrially using an enzyme, you don't want an inhibitor. You don't want a good inhibitor. You don't want an inhibitor with a Km lower than the Km of the substrate.
Got it. Yeah.
Okay. So another type of inhibitor is non-competitive inhibition. And in this case, the inhibitor doesn't bind to the active site. So it's really not influencing the affinity of the active site um, to the substrate. What it does, it binds at a different location. And when it binds to that different location, it then slows the rate, or it will slows the rate of the reaction because the substrate no longer is able to bind. So once it binds, it changes the configuration, and then the substrate doesn't bind. The rate of the reaction is reduced because some enzymes have the inhibitor, some enzymes don't. So it lowers the available enzyme that are reacting with the substrate. So it lowers the production of the product. So basically, it lowers the rate of the reaction. So here's nothing happens to the Km. However, there is a decrease. Remember, this is an inverse 1 over V0. So this higher value here indicate as a lower initial velocity. So basically, it reduces the rate of the reaction, but does not influence the Km. Another form of inhibition is when you have the ES complex, the inhibitor binds to the ES complex. So it doesn't bind to the enzyme alone. Once the enzyme and substrate are bound, it binds to the complex and then prevent the reaction from going forward. In this case, you have higher Km as well as lower velocity, and you end up having the same slope because they changed proportionally. Um, yeah, so but basically slower rate of the reaction overall and higher Km, but if we look at the slope, it will be the same, but you have different intercepts and then you have different Km. So this is recap questions, but I'll keep moving on so that I can finish the lecture. But Radhika, if you want to leave and you're tired, you can too. But if you want to stay, you're welcome to stay. I'll just continue. These are just recap questions based on what I just covered. So just make sure that you review them and you know what they are when you're studying. Okay, this is example of an application to determine whether the inhibitor is a reversible, what type of a reversible inhibitor we have in the system. So this is also work I've done as a postdoc at Purdue. We were looking at enzyme system in milk, natural enzyme system in milk. Plasmin is the enzyme that hydrolyzes beta casein, which can cause problems during storage of milk, but is desirable during cheese fermentation or cheese aging. So we needed to understand the system in milk. In the same system in milk, you have plasmin, you have plasminogen, which is the inactive form of plasmin. You have plasmin activators, plasminogen activators that produce plasmin, and then you have plasmin inhibitors as well. So in order to understand how do we treat milk so that we can either inhibit the inhibitors, inactivate the inhibitors, or inactivate the activators and keep the inhibitors, depending on the final application of milk. So we need to understand the system basically. So we wanted to know what type of inhibitor is the inhibitor present in milk for that enzyme. So we again isolated the plasmin and isolated the inhibitor from the milk, and we prepared the reaction with and without the inhibitor at different concentrations, which is a substrate similar to beta casein, but synthetic substrate that would react with plasmin and give a product that we can measure with spectrophotometry. So when we did the assessment, first when we plotted, we noticed, oh, we have similar intercepts. So this tells us it is a competitive inhibitor from just looking at what happened here. So we increased the slope, but however, the velocity was not impacted. So we have similar intercept. And when we actually got the equation of the lines and calculated Km, we notice an increase in Km. So you go from -2 to -1 factor. So this means an increase in Km in the presence of the inhibitor. We didn't see a significant difference in Vmax, so we had similar intercept. We calculated Kcat, which is Vmax over total enzyme concentration, because Vmax is the same. It didn't, Kcat was the same, but the catalytic efficiency, when you take Kcat over Km, it got reduced because Km increased. So the catalytic efficiency overall in the presence of the inhibitor was reduced due to the increase in Km. All right, so we determined this is a competitive inhibitor. So when we are looking at inhibitors choose when we want to prevent a certain reaction in food system, and this is an example for polyphenol oxidase. And then what inhibitors we want to use. Do we want to use a competitive inhibitor, or do we want to use a non-competitive inhibitor? So in most cases, you want to use non-competitive inhibition, because with competitive inhibitor, if you have high substrate concentration, it's not going to do anything. So it's best to have a competitive, non-competitive inhibition in this case. But what you consider is the cost, how much is going to cost me to add a sufficient amount of that inhibitor. Is it safe to use? Is it FDA approved for food use? Does it have any sensory impact? So we always have to consider other things, not just inhibiting any reaction, but other things that go into deciding on the inhibitor. So measuring of enzyme activity, or measuring, well, let's start with measuring of enzyme activity. In this case, you would hear about when you buy an enzyme, you might read units of activities per gram of protein. So there is a difference between enzyme activity and actual concentration, basically how much enzyme you have or how much protein you have. So you often get activity per gram of protein or per certain amount of your mixture, if it is in liquid form or powder form. So what is the unit of activity? So there are different, it's not universal, because every enzyme have several substrates they can work on. And units of activity is really specific. They're determined based on a specific substrate. So usually they give you a unit of activity, let's say micromole per minute, but it is specific. So micromole, so unit of activity, let's say 1 unit of activity for an enzyme is potentially equals to 1 micromole of substrate converted in 1 minute at a particular pH and temperature. So, because the activity of enzyme is impacted by so many factors, the substrate, the pH, the temperature, cationic strength, so it is very specific. So when you read the unit of activity of one enzyme and you read units of activity of another enzyme, but you cannot compare because you have to see what substrate was used and what conditions were used. So that is something that you have to pay attention to if you are buying enzymes and you're trying to determine, understand the units of enzymes or unit of activity. So oftentimes enzyme assays are coupled reactions. So we have the first reaction, which is the measuring reaction, where you have, let's say, either your enzyme of interest or substrate of interest, and you get a product, but you can still not get any absorption. Neither of the substrates or the products have UV or any visible or UV absorption. Then you need to react the product with another substrate and another enzyme to get to a product that you can measure spectrophotometry. So each enzymatic reaction obviously needs its own pH and optimum temperature. And we try to link to choose enzymes that have similar pH and similar temperature optimum, so you don't have to change within an assay to change pH or change temperature. So two things we need to consider is the pH of the reaction for both enzymes to be similar, and you want the second reaction not to be limiting. That means you want it to go fast, not to be delayed. This is an example of a coupled reaction. In this case, we are measuring glucose. So we are after glucose determination using enzymes. So in this case, for example, we have hexokinase, which is a transferase. So basically it transfers a phosphate from the ATP to glucose to get glucose 6-phosphate and ADP. So neither of those are going to give you a UV or a visible detection. So you want another enzyme. You take the product from here, add an ADP, to the dehydrogenase enzyme that requires this as a cofactor. And what we get is the NADPH, which absorbs UV light, and we can get the detection, and using standard curves we can get the concentration of glucose. So the last section of this is of the enzymes is really looking at two main categories that are influential in food space. So we have the hydrolyses and we have the oxidoreductases. So we're going to talk a little bit about hydrolyses, examples here, and applications in food, and then oxidoreductase. So hydrolyses, so what's the definition basically? The definition of the hydrolase is where water is a part of the reaction. So you use water and then the enzyme and the components of water gets attached, taken up in the reaction. So for example, if we have triacylglycerol and we use lipase, it breaks off one fatty acid in presence of one water molecule, and you got diacylglycerol, and the water components is part now of the diacylglycerol. So that's why it's called hydrolase. So there's the hydra, there's the water. So the water is part of the reaction. And hydrolase breaks covalent bond. So it breaks covalent bond, and then the water components are taken up by the compound. So pectinases are very important in industry. We use them for different purposes, and sometimes we need to inactivate them. So pectin, used in industry for different purposes as a gelling agent, add viscosity, and also stabilizes. So it sometimes specific pectins are added, for example, in high protein beverages to stabilize the protein in solution. So they have different applications. And we have a commercial low methoxypectins. That means, so these are the glucouronic acids. So you have methyl group sometimes on some units, and other units don't have that methyl group. So these, in this case, you have here methyl group and you have a methyl group, but there are units that don't have the methyl group. It's just the acid, COO8, COO minus or COOH, depending on the ionization state. So you have low methoxypectin, less than 50% esterified. So that means less than 50% of the units have methyl groups. They form thermoreversible gels when you add calcium ions. But if you have a high degree of esterification or deesterification, so you can reduce it. So you can reduce the esterification. You can use pectin methyl esterase that would take off the methyl group, and you have more COO minus. So you end up having more charges, negative charges, and in the presence of calcium, the more negative charge you have, the more cross-binding you're going to get between the polymers. So stronger the gel, the firmer is the gel. So basically, you can enhance pectin from gel formation from pectin by reducing the methyl groups by deesterification of methyl groups. But so I'll show you an example when it is not desirable to do so, though. But first, we have also the pectinases that break the bond between two glucuronidase acids. So you have the polymethyl galacturonase that breaks a bond when you have a methyl group. And you have the polygalacturonase, which breaks the bond between two units that do not have a methyl group. So, okay. So the example I give you here is clarification of apple juice. So when you press the apple, you have lots of pulp. And if you want to clear apple juice, you add pectinases. So you break these bonds into singular uronic acids, galacturonic acid that are soluble. So then it's a way to clarify apple juice. Now, in this example, the pectin methyl esterases that are naturally present in orange juice can cause this to happen. So if you have an orange juice that is fresh and then you let it sit, the pulp separates. So if you want to have an orange juice, to produce orange juice and package it, and you don't want the pulp to separate, you want to make sure you inhibit the endogenous pectin methyl esterase. Because what they're going to do, the pectin methyl esterases are going to break off the methyl groups and create more charges. And in the presence of divalent positive ion like calcium, they're going to bind together, form big polymers, and precipitate. So your pulp is going to precipitate and separate from your juice. You don't want that to happen. So orange juices are usually thermally treated, especially if you want to maintain that thickness of the pulp without separation, to inhibit pectin methyl esterases. So here's a case study where they're looking at inactivation kinetics of pectin methyl esterases in orange juice, where they don't want it to treat the orange juice at high temperature because it changes the flavor. So they're looking for ways to treat the orange juice in a way that you don't have to heat at high temperature to inactivate the enzyme and affect its sensory properties. So they looked at high pressure combined with some thermal treatment, and they did look at inactivation kinetics. So what they found here, this is the table, they looked at rate constants and they looked at the D value, which is the 1 log inactivation of the enzyme, like 90% reduction in activity. Okay, so they found in this case, they looked at temperature combined with pressure, and they found, yes, at higher pressure, we can get uh, a very low D value, which is the minutes to reach 1 log reduction, basically. So they found that a combination of 650 degrees gets you really good inactivation, but even 25 degrees get you very significant inactivation compared to here, for example, 117 minutes to 7 minutes. That's a great reduction at same temperature. Okay, so other forms of hydrolysis are amylases. So we have alpha amylase, we have beta amylase, glucoamylase, and polynase. So basically, they work on starch or maltodextrin molecules. If we look here, I always show this. I guess I took it from a former colleague of mine, um, Mirko Banzel. Before George, he was our carbohydrate chemist. So I stole this from him. I like this slide. It just shows where the different enzymes cut and which ones are exo and which ones are endo. That means if they're exo, they're basically from the terminal, they're taking units from the terminal ends. If they're endo, that means they chop from the middle of the molecule. So glucoamylase takes one molecule at a time from the terminals. So it takes one glucose unit at a time. Alpha amylase is an endo, so it kind of cleaves, mostly it cleaves the alpha 1 4 linkages, but it does also cleave the alpha 1 6, which is the branched. Polynase is specific for the one alpha 1 6. It's an endo and it cuts specifically at the alpha 1 6. So if you want to deep branch, polynase is a good enzyme for that. And then you have the beta amylase. It's also an exo enzyme, but it cuts 2 to 3 units at a time from the terminal ends. So what is an application? An application, for example, is high fructose corn syrup. So we start with starch, add alpha amylase, we get maltodextrins, we add glucoamylase. It breaks the maltodextrin more into glucose singular units, and from there we use an isomerase in this case to convert to fructose, glucose to fructose, and there you have the high fructose corn syrup, which came from starch and became sugar. Lactase
And I
is,
have a question in the
yeah.
previous slide. We could also use beta amylase instead of alpha amylase, right? Like, is it specific?
It's slower. If you want to use beta amylase, it's gonna, chop 2 3 units at a time, and I don't think it is specific potentially to alpha 1 6 linkages. So usually when you want to break the starch molecule, you want to use alpha amylase. It's more efficient to get you smaller units, like to get you to hydrolyze and get you smaller maltodextrin that you can then use the glucoamylase. It's very rare that you would use beta amylase for such an application. I don't think anybody does that. Yeah. Okay. Right. And then lactase is commonly used for example, to produce delactose milk for lactose intolerance. So it basically breaks into glucose and galactose for those people that cannot hydrolyze lactose in their gut. So that is used industrially. Sucrase is also used industrially, also known as invertase. A very common application is the the chocolate coated cherry. So what happens is they form the syrup And then with sucrose, it's more viscous and crystalline. They dip the cherry in it and then they coat it. Uh, Over time, so you want that syrup to be liquidy and not crystalline. So over time, inside of this chocolate coated, then the invertase convert sucrose to glucose and fructose, and they have lower crystallization potential. Then when you bite into it, you have that nice syrup that comes out that is not crystalline and it's smooth. So that's an application. Proteases have so many different applications. One for texture or changes in texture, so curd formation, for example, or tenderizing of meat. To make the meat more tender, sometimes you add bromelain enzyme or papain enzyme to the meat cuts to make them more tender. Um, Disruption of gels. Proteases can break down the proteins, disrupt gels, are important in aging of cheese. They produce the flavor. You can produce hydrolysates for enhanced activity, bioactivity, or functionality. And sometimes you can add them to wheat gluten to make the gluten more viscous than elastic. So you can break up the large molecules. So you end up with more viscous, a more extensible kind of end product rather than too elastic. You enhance extensibility versus elasticity. So different applications, different enzymes, just a table that shows some of the enzymes, and some are different in specificity, some are general, some are more specific. For example, trypsin is very specific to breaking after lysine or histidine. Others, pepsin is very general protease. And then, of course, all of them, most of them should have EC numbers. All of them should have. I don't know why the table is missing some of that. But some enzymes are characterized as metalloenzymes because they need metal group or element to enhance their activity in the active site. Some are serine proteases, they call them, because they have a serine group in the active site. So if you see carboxyl or serine, that means in the active site they have a specific carboxyl group or serine group or cysteine group or metallic group. Um, Lipases are naturally present in dairy products, and they aid in formation of flavor during ripening. They are added to enhance emulsification, so to produce mono and diacylglycerols. So this is a monoglycerol, diacylglycerol. So there are emulsifiers used to are added to emulsify in different applications. The last group I'm going to talk about is oxidoreductase. So what's its definition? It catalyzes a reaction where there is either an oxidation or a reduction of the substrate by transfer of hydrogens and electrons or by use of oxygen. So here in this case, catalase or also peroxidase reduces hydrogen peroxide into water and oxygen. So this is a reduction reaction where you get rid of hydrogen peroxides. Um, the most common enzyme of problematic in industry is polyphenol oxidase. Actually, a large percentage of produced vegetables and fruits are lost because of polyphenol oxidase. A large percentage, I can't remember what the percentage is right now, but it's really large percentage. I was surprised when I saw it. But anyway, so it causes browning. Sometimes it's desirable, like in tea. Sometimes it's not desirable. You don't want to buy a shrimp that looks like that. Maybe it is not bad for our health, but it's potentially not good looking. And also produce. So you have this browning that would impact the quality and therefore they're lost in terms of nobody wants to use them or buy them. Um, they have the bioengineered apples, for example, the Granny Smith. Why don't they brown? Because they're bioengineered to have low concentration of polyphenol oxidase, so they are more slower to brown when you cut the apple. So what's the chemistry behind the browning reaction with polyphenol oxidase? So what happens is you start off with a monophenol, and when it's exposed to oxygen, when you cut the apple, cut the potato, or whatever, you're exposing it to oxygen, then the endogenous enzyme, you have two different types of enzymes, serisolase and catecholase. One will convert the monophenol to diphenol, so you get two phenolic groups. And further oxidation in the presence of oxygen gives you a quinone, which is a browning pigment. So what it does, it can react with other polymers, such as amino acids and proteins. And you get oxidative condensation because it's an oxidant. And then you end up with large brown polymers, which are known as melanins. So we can actually reverse this. So we can reverse the this reaction by adding a reducing agent. So this quinone can be reduced back to diphenol in presence of reducing agents. Ascorbic acid is a reducing agent. Thiol compounds such as cysteine, sulfides such as sodium bisulfide, um, are reducing agents. So they basically reduce back the quinone, prevent the polymerization from happening. Um, there is also treatment with sulfur dioxide, which is also is added to prevent browning in dried fruits and such. So what it does is basically causes it reacts with the quinone and produce a sulfur adduct. This sulfur adduct is not capable of producing oxidative condensation and melanins. So it kind of inhibits the progression into melanins reaction. So there are so many methods that have been investigated to inhibit this polyphenol oxidase because of its impact on the loss uh, of these produce. So heat inactivation, but you don't want to eat the cooked apple. Dehydration, not always you want to eat a dried fruit or vegetable, but still it kind of reduces the browning. Making it acidic, adjustment of the pH, so oxidation is not favorable at acidic pH. Reducing agent, chelating agents that would catch up methyl group that would enhance oxidation. Aromatic inhibitors, so these act as competitive inhibitors, for example, so benzoic acid, cinnamic, ferulic, they can bind to polyphenol oxidase instead of binding to the phenol substrate. So they would inhibit the reaction by being competitive inhibitors. Modified atmospheric packaging, where you change the atmosphere from being high in oxygen to low in oxygen and maybe high in argon. I'll give you an example here in a minute. Genetic engineering of uh, the PPO gene. So basically, that would result in reduced production in the crop or in the fruit or vegetable of PPO. Irradiation and high pressure processing. So this is an example where they looked at enzyme kinetics to compare two different enzymes from two different sources um, of peaches, two different varieties of peaches, to determine their affinity to different substrates. So to understand the differences of these enzymes, which are more potent, and they wanted to look at Km and Vmax. And they looked at substrate specificity to different substrate and inhibition by different inhibitors. So what they looked at these two different enzymes And they looked at different substrates, and they found that different enzymes have different affinities to the different substrate. So for this variety, the first variety, it has the lowest Km to this particular substrate, whereas the second variety has the lowest Km value and the highest catalytic efficiency for this other substrate. So two different sources of enzymes react differently to different substrates. Okay, and then they looked at inhibitors and determined the type of inhibition, mostly competitive because they don't see much change in Vmax. And they determined which inhibitor actually reduces the reaction the most for the two different types of enzymes by looking at the reaction rate constant in this case. So they looked at the reduction in the reaction rate constant. Okay. The second study, which is the last one, they looked at changing in the packaging environment and looking at the kinetics of that. So oxygen is a substrate, right, for PPO. So they tried to replace oxygen by argon. So they looked at argon, changing it to mostly argon and a little bit of oxygen, and looking at increasing the concentration of nitrogen and reducing the concentration of oxygen. So they looked at air, where I believe the concentration of oxygen is about 28-30% maybe, and the rest is mostly nitrogen. So for that, they looked at two different types of PPO from different sources. They noticed that the Vmax in any of these cases did not change. So the Vmax remained the same, but the Km changed. So it actually increased in some cases, and they found that argon, substituting with high percentage of argon, is most effective in increasing the Km and reducing the affinity to the oxygen substrate. So, and then in this case, they saw very much increase in Km for this particular enzyme. So another application of kinetics to understand active packaging, packaging, changes in atmosphere. So which type of inhibition? Obviously, I just kind of answered it. You have similar Vmax, different Km. So it's basically competitive inhibition. I lied, so we still have glucose oxidase. So this glucose oxidase is used in food analysis for detection of glucose, quantification of glucose. And it requires two enzymes. So glucose oxidase produce hydrogen peroxide and gluconolactone. The hydrogen peroxide is used to oxidize a dye in the presence of peroxidase. And then the peroxide is reduced and the dye is oxidized, basically. And then you can measure the absorption and relate it to glucose by standard curves. Glucose oxidase in industry is used as oxygen scavenger in bioactive packaging. So they immobilize glucose oxidase in the package, and then it kind of scavenges oxygen, so prevent oxidation of lipids or colors or flavors. Okay. Application of immobilized enzymes. So we have in industry, they immobilize the enzyme so to enhance the efficiency of the use so that they're not lost in batch processing. So they immobilize them on membranes or reactors. And this is used to produce corn syrup for going through to make the milk free of lactose, so pass the milk through reactors where you have immobilized lactase or beta galactosidase, the same as lactase. Production of protein hydrolysate. You also have you have immobilized them on membranes that are porous. You have your proteins. They get hydrolyzed, small molecular weight hydrolysate pass through the membrane, and everything else is becomes retentate and removed, and the enzymes remain in the membranes and continuous process. And of course, enzyme linked immunosorbent assays where the enzymes are immobilized either on the antibody or on the antigen for application, ELISA applications. So here's our examples of enzymes in different food systems. In baking, we use amylase to enhance the fermentation process and reduce the staling because it breaks down some starch, so reduces the retrogradation effect, formation of large polymers. Proteases, we talked about several uses, but in baking, you can reduce the elasticity of gluten, enhance rheological properties. Um, the That's the glutamyl transferase, basically that enhances polymerization. So when we start with wheat that has low strength of gluten, you can enhance that, or any flour that has low viscoelastic properties, such as rice flours, you can add glucoamylase. No, what is it called? Not glucoamylase. What's the other names of
Contaminase?
glutaminase? Yes, thank you. Yeah, here they call it glutamyl transferase. I never used that, so it made me forget what the original name is. Sometimes you can oxidize sulfidyl groups, so you can add an oxidizing agent. So, for example, they add ascorbic acid. Well, yeah, for example, they add ascorbic acid to flour to enhance the formation of disulfide linkage. In this case, they add an enzyme which induces disulfide formation. And this is the transglutaminase example where a rice flour is now able to form a loaf bread without collapsing by just enhancing the polymerization. And here you have the amine group of a lysine here with the carboxyamide group of glutamine. And that's the transglutaminase forming polymer. In milk, chymosin for cheese, proteases and lipases for flavor. Um, well, formation of disulfides actually remove the sulfidyl kind of taste, the cooked flavor taste, removing of lactose, and well, this is soybean. I don't know why it's in milk and dairy, but um, enhancing coagulation in soy milk. Microwave protease shouldn't be in this table. I'm not sure why it's in this table. And sometimes use enzymes to remove unwanted constituents. Again, the lactose, hydrogen peroxide by catalase or peroxidase, debittering peptides by exoproteases or general proteases. You can break down the peptides that are made up of a lot of hydrophobic amino acids, break them further, you debitter them. And then to get rid of protease inhibitors, and these are proteins, you can use proteases to break them as well. Analysis, different constituents, sulfites, so that we can make sure to label our products for sulfite sensitive individuals. So we need to measure the amount of sulfite in certain products. Glucose, we need the enzyme, starch, we break it all the way to glucose, measure glucose, and then back calculate the amount of starch. D-malic acid uses an enzyme to, this D-malic acid is synthetic form of malic acid that is present in apple. Natural is L configuration. And if that D-malic acid is present, that means it's synthetic product. And then measuring enzymes, peroxidase and lipoxygenase. Usually when you blanch fruit, vegetables, you want to deactivate enzymes, and these are the most thermally stable enzymes. So you measure one of them, and if it is inactivated, then the blanching is um, um, done properly. Sometimes we monitor lipoxygenases in grains and legume seeds because endogenous lipoxygenases causes oxidation of fat and reduces shelf life. Alpha amylase is important to measure in flour. High activity can cause starch to break down and lower viscosity in products. Measuring the activity of rennet, for example, helps us determine how much rennet we use for how much amount of milk to produce to enhance our curd production. And that's ended. Thank you, Radhika. You're the sole soldier that is here. But yeah, that's all for today. I will post that for everybody else. Do you have any questions, Radhika?
No.
All right. Thank you for being here and see you later.
See ya. Have a good evening. Bye bye.
Bye.